This lecture will talk about the plasmid purification experiment. We use plasmids in the biotech industry by putting genes in them that code for useful things that we want. Examples of how plasmids have been used to engineer useful genes in things are allowing plants to grow in increased salt environments, or having ampicillin resistance in certain areas. So in any way you want to do it, you have a donor plasmid and you have a recipient plasmid. The donor plasmid contains the gene of interest, and next to the gene of interest are generally some restriction enzyme sites that I'll mention again in a moment that you can use to, you have a restriction enzyme cut the DNA fragment out of the donor plasmid. At the same time, you are cutting the recipient plasmid with the same restriction enzymes. The recipient plasmid generally has what's called a multiple cloning site in it, which has a whole bunch of different restriction sites all very close to one another. So no matter what restriction sites you are using for the donor plasmid, you're going to be able to find something that will work to be able to put it into your recipient plasmid. So you cut out the fragment, you digest with the restriction enzymes, cut it out, and you insert it in using a chemical called ligase to ligate your fragment into your plasmid of interest. Now, this is not how our plasmid looks. We do not happen to have a neomycin-resistant gene in our plasmid, but we will have an ampicillin resistance gene in our plasmid. And in one plasmid we will use later in the semester, we will actually use an engineered plasmid which codes for a protein of interest that we want to be producing. This is the way that insulin was produced. Years ago, when someone was a diabetic, they had to take insulin which had been isolated from pigs. But biotech companies found the gene that coded for insulin, and they were able to insert it into a plasmid and put it in bacteria, and then large amounts of insulin could be produced, and the price of insulin dropped dramatically, allowing many more people to survive having diabetes. So this is an example of how this can be used for good to help humanity. Once you have your plasmid of interest, you have on that plasmid an origin of replication. We'll be talking about that later. And you have your specific antibiotic resistance gene on it, as well as whatever you've put into your plasmid. And the antibiotic resistance gene is important to have on the plasmid because you need to be able to put it into bacteria and only have the bacteria grow that contain your plasmid. If you make a plate that contains agar, allowing bacteria to grow, if you were simply to spread bacteria across that plate, you'd have millions and millions of bacteria growing on that plate. So you need to be selective. You need to be able to pick which bacteria are going to have my particular plasmid. And you do this by putting an antibiotic resistance gene into the plasmid. So when that plasmid is in a particular bacteria, then the bacteria are plated onto a cell which contains that particular antibiotic. So if you have an ampicillin resistance gene, you'd put it onto an agar plate which contains ampicillin, and then only the bacteria which contain your plasmid will be able to survive on the plate. The trouble is, how do you get the plasmid into the bacteria in the first place? And there's several ways to do it. You can use calcium-mediated transformation to get it into the cell. Basically, you take your bacteria and you mix it with a calcium phosphate or a calcium chloride solution if it's bacteria, calcium phosphate if it's tissue culture cells, and this forces little holes to open in the bacteria, and this allows the plasmid to slip in. Another way of getting a plasmid in a cell is electroporation. You put it into a special cuvette, which sends a jolt of electricity across all of the cells, again, making little holes in the bacteria and allowing plasmid to slip in. And in both cases, you need to be able to allow the bacteria time to heal up those holes and grow and survive. If you're working with tissue culture cells, another way is lipid-mediated transfection, where you take lipids that sort of are going to have the same consistency as a membrane for tissue culture cells, and you mix those with your plasmid, and this creates little micelles, which then grab onto the membrane surface and are incorporated into the membrane surface and allowing plasmids in cells. So there's different ways of getting plasmids into the cell, and different depending on whether you're doing tissue culture or whether you're doing bacteria. But basically, you need to have some way of telling which cells actually contain the plasmid, ensuring that only those cells are allowed to survive. So assuming that you have your plasmid in the E. coli cell, there are definite size differences between the E. coli genome and the size of a plasmid. Now, plasmids can range in size from 2,000 to 20,000 base pairs. Our plasmid we're going to be growing is around 3,000 base pairs, so it's on the small side of that, particularly compared with the E. coli genome, which is exceptionally large, 6 times 10 to the 6th base pairs. And I need to note right here, because it's going to be important later, that the E. coli genome at one point is attached to the cell membrane, and I'll explain later why that's important. Plasmids, on the other hand, are able to replicate independently of the E. coli genome. Plasmids use the polymerases that are naturally found in the cell inside the E. coli, and they're using those polymerases to be able to replicate themselves. So plasmids are generally containing non-essential genes under normal growth conditions, but remember, as soon as we add ampicillin to the culture where we're trying to grow our E. coli, then all of a sudden this ampicillin resistance gene allows the E. coli cell to survive only if it contains the plasmid. If it does not contain a plasmid, this E. coli will die. Plasmids are the foundation, as I said, of recombinant DNA. And I mentioned cutting with restriction enzymes. Restriction enzymes can cut the DNA in different ways. So if you had the sequence GGATCC and you had BAMH1, which is the name of a particular restriction enzyme, BAMH1 tends to make a nick in the DNA right here and a nick in the DNA right here. So when you separate the DNA, it's going to look like this. So in this case, it has a 5' 4 base overhang when it's cut with BAMH1. When you cut it with PVU1, on the other hand, it's going to nick the DNA right here and right here. And when you separate those pieces of DNA, in this case, you have a 3' overhang. So in this case, you would have a 2 base 3' overhang to the DNA. Because remember, DNA is 5' to 3' in this direction and 5' to 3' anti-parallel on the other strand. Now, sometimes you have enzymes that will be blunt cutters, and if you have an enzyme that's a blunt cutter, there'll be no overhang at all. So when you are trying to ligate your your fragment into the plasmid, it's important to have restriction enzyme sites that are compatible. You would not be able to ligate this end to this end right here because those bases would be in the way. You would need to find a way to get rid of them, for example, or to use a different restriction enzyme right here. So restriction enzymes generally are cutting double-stranded DNA at a specific sequence that's recognized by that particular enzyme. So if you have something that recognizes a 4-base restriction enzyme sequence, GATC, in this sequence, GATC, antiparallel in the other strand of DNA, so it's a palindromic site, you would have GAs, T's, and C's that would have a 4-base recognition site, so it would cut every 4 to the 4th or every 256 bases in your large plasmid that's 2,686 base pairs. So you can imagine if it's cutting every 256 base pairs in this large plasmid, you have a lot of DPN1 restriction sites in there. On the other hand, if you have a 5-base recognition site, it would cut once every 1,024 bases. If you have a 6-base recognition site, it would cut once every 4,096 bases. So BAMH1, for example, has a 6-base recognition site, GGATCC, and then the palindromic GGATCC on this, so it reads the same in both strands of the DNA, and that's what the restriction enzyme is actually recognizing. So if it cuts once every 4,096 bases, it is very possible to have a bunch of places on the DNA where you only have a single recognition site for that particular enzyme in the entire plasmid. And that's what makes this multiple cloning site right here so useful, is that if you need to cut with BAMH1 and small 1, or KPN1 and APO1, or HIND3 and SPH1, you will find all of those restriction enzyme sites in this very small area of the DNA. Now, PUC19 actually happens to have a LAX-Z gene in it, and that's used for blue-white selection, because if you have a fragment cloned in right here, you are disrupting the activity of this gene, and if you have a substrate added, which can cause a blue color in the presence of the gene product, if you interrupt the gene, the colony will be white because it will not be making that blue color. So this is called blue-white selection. Not every plasmid has it, but many plasmids do. We're going to be growing PUC19 in class, and this happens to have blue-white selection, although that was already done for you when things were cloned into the plasmid. So here we can see our ampicillin resistance gene along with an origin of replication. When you are growing bacteria, there's generally a lag phase in how many cells you are able to exponentially grow. So generally bacteria are very good at hanging on even when there's no nutrients in the solution. So when you take bacteria and put them into a new solution with a bunch of nutrients in it, it takes a while before they will start growing. But eventually they will start growing logarithmically in using the nutrients that are in the solution. And again, as the solution runs out of nutrients, they go into a stationary phase. They're not dead, but they're not actively growing either. And it's only if you don't give them new nutrients that they go into a decline phase where the cells are actually they're declining in numbers because they're running out of ways to keep their membrane intact or things like that. So then they will actually die. So you want to be growing cells in a logarithmic phase, an actively growing phase, when you are adding certain chemicals to them that you need to have effective on the bacteria. Now, as I said a moment ago, our PUC19 plasmid has an ampicillin resistance gene on it. Now, the mode of action of ampicillin, when you have ampicillin in the culture or in the agar plate, ampicillin is inhibiting cell wall biosynthesis by interfering with the peptidoglycan cross-linking necessary to make the cell wall of the bacteria. So if you have ampicillin in the culture, if you have a bacteria which does not contain the plasmid with the ampicillin resistance gene, the bacteria is going to die because it's not able to make the cell wall properly. On the other hand, if you do have a plasmid in there, the mode of resistance of ampicillin is it makes an enzyme called beta-lactamase. So that ampicillin resistance gene codes for beta-lactamase. Beta-lactamase cleaves the beta-lactam ring of ampicillin, and ampicillin is a type of a penicillin. So when you cleave the beta-lactam ring, you are inactivating the ampicillin, so it's no longer able to inhibit cell wall biosynthesis. So we're going to have ampicillin in all of our cultures. That's one antibiotic we're using in this experiment. Another antibiotic we're going to be using in this experiment is chloramphenicol. The mode of action of chloramphenicol is that it inhibits the translation of the 50S ribosomal subunit at the peptidyl transferase step. In other words, no protein can be made. If you add chloramphenicol to a culture where you have bacteria growing, you will stop the production of protein of those bacterial cells. Now, if you happen to have a chloramphenicol resistance gene, what the chloramphenicol resistance gene does, it causes acetylation of the chloramphenicol by chloramphenicol transferase. That's what the gene codes for. But we do not have, let me say that again, we do not have a chloramphenicol resistance gene in our plasmid. So when we add chloramphenicol to our growing culture of cells, it's going to cause protein production to stop in our bacterial cells. Now that's important because E. coli replication requires protein production. And as the genome for E. coli is made, in the genome, there are early genes in the E. coli genome, and those early genes serve to turn on the middle genes, and those middle genes serve to turn on the late genes. So if you have no protein being made, the gene product from the early genes can't turn on the middle genes, and the middle genes cannot turn on the late genes. So if you add chloramphenicol such that you have no more protein being made, E. coli replication is stopped. No more cells are going to divide. But here's the interesting thing about that. You already have proteins which are in your bacterial cell. No new proteins can be made, but there are other proteins like polymerases that are still floating around. So the plasmids are able to use those polymerases which are already in the E. coli floating around. The plasmids are able to keep replicating. E. coli cannot replicating because they require fresh proteins to be made with every round of replication, but the plasmids are able to use the existing proteins, the existing polymerases, to make multiple copies of themselves. So what you basically get in the cell is you get multiple copies of the plasmid for every single cell that you have. So you're basically increasing your yield of the plasmid. So then, you need to take all of your cells and you're going to spin down all of your cells after they have been with the chloramphenicol overnight, basically. You can't leave the chloramphenicol on too long because eventually the cells will start to die if they're not able to replace the proteins that are in the membranes, for example, that are keeping them intact. So an overnight shaking with chloramphenicol is what needs to happen, and then the cells are spun down and you get a cell pellet. So in the cell pellet, basically what you're doing is you are disrupting the cell membrane and getting a cell extract out of that. And then from your cell extract, you are centrifuging to remove, to separate out the DNA, the RNA, and the protein from the membrane. Now, remember that I mentioned that the E. coli genomic DNA is attached to the membrane at one point. So this is important because when you spin down the membrane of the cell, the E. coli genomic DNA is going to go with it and be in the pellet. And what's going to be in the supernatant? The plasmid DNA, the RNA, the protein, and any broken genomic DNA. Because the E. coli genome is fairly large, and you're swirling your sample around, so it's possible you might have broken up a little bit of that E. coli genomic DNA. But the majority of the E. coli genomic DNA is going to be down here in the debris. You might have some E. coli genomic DNA along with your plasmids in the supernatant. So what you're going to do is you're going to bring up your pellet in a 25% solution with sucrose and tris. Tris is a buffer. You're doing sucrose because you want an isoosmotic solution. You don't want to prematurely break your cells. And to those cells which you're bringing up in a solution, you're going to add lysozyme. And lysozyme starts chewing away the cell walls, exposing the membrane to the solution. And in that solution, you're then going to add EDTA. Remember that EDTA chelates divalent cations to reduce the protease and DNase activity. We're not so interested in proteases this time, but we're very interested in DNases. So we want to make sure that we have sequestered all those divalent cations so that they cannot, so that all the DNases are inactivated. So now you have E. coli cells that you've added lysozyme to chew away the cell wall so that the membrane is exposed, and you're going to add Triton detergent to it. And that's going to cause all of your cells to break open. And then you're doing that differential centrifugation, which is going to separate the membranes and the intact genomic DNA from the other cell components. So in the pellet, you're going to have your membrane and your intact genomic DNA. In your supernatant, you're going to have proteins, RNA, plasmid DNA, and broken genomic DNA. So then you're going to bring your total volume to 20 mils of solution, and you're going to add 20 grams of cesium chloride to that. And the reason you're doing this is you're going to be doing a density gradient. And that density gradient is going to separate these compounds by their density. Not by their size anymore, but by what density they are. So if you have 20 grams of an aqueous solution and you add 20 grams of cesium chloride is an exceptionally dense salt. You will see that the total volume there is only 25 mils, or your average density in this case is going to be 1.6 grams per mil, which is about the same density as DNA. So protein has a lower density, RNA has a higher density than your DNA does. But at this point, DNA is DNA. So the question is, well, how are you going to separate plasmid DNA from E. coli broken genomic DNA? And the answer is ethidium bromide. Ethidium bromide intercalates into DNA. It intercalates into all DNA. It's a flat planar molecule, and it's somewhat hydrophobic, so it does not like being inside, it does not like being in an aqueous solution. And so because it's a flat planar molecule, it can push its way in between the bases of the DNA. When it does that, it untwists the DNA slightly. And this changes the density of the DNA. So now that the ethidium bromide has intercalated into the DNA, it's making your DNA less dense. And so changing the DNA density, different rate than the broken genomic DNA than the plasmid DNA. So why is ethidium bromide able to intercalate differently into broken genomic DNA than plasmid DNA? Well, plasmid DNA is a covalently closed circle. Its ends are not free. So a very limited amount of ethidium bromide can intercalate in there because the ends are not able to freely twist. Compared with broken genomic DNA, you can have more and more and more and more ethidium bromide intercalating in there because these ends can simply keep untwisting and untwisting and untwisting, whereas the plasmid DNA cannot, so you can only intercalate a little bit of ethidium bromide into plasmid DNA. So plasmid DNA will become a tiny little bit less dense, but the broken genomic DNA will become significantly less dense because it can intercalate more of the ethidium bromide. So then you're able to see the difference on a gradient and separate out your plasmid DNA away from your RNA, away from your RNA, and away from your broken genomic DNA. Now, you need to extract that ethidium bromide out with an organic alcohol to avoid damage to the DNA. So you need to basically get rid of the ethidium bromide as soon as you have separated away from the plasmid DNA. So this is kind of what it looks like. You have a gradient with your supercoiled DNA, and that's going to have a different density than your linear DNA, and it's going to have a different density than your protein and then your RNA. We're going to be using a syringe to separate out specifically the band with the supercoiled DNA. And then we're going to be adding, after we do that, we're going to be adding isoamyl alcohol. This says N-butanol, but we're going to be adding isoamyl alcohol and vortexing it together. Then the ethidium bromide will go into the organic phase, and it will actually turn sort of pink, and the DNA will be in the aqueous solution, and the aqueous solution will turn from pink to clear as you extract the ethidium bromide out. Just like when you were in organic chemistry and you had to extract a couple times, we'll have to do a couple of extractions to make sure that we have all of the ethidium bromide out of our DNA solution. So then you need to precipitate down the DNA. And to precipitate DNA, you need to have some salt in there. Cesium chloride, of course, is a salt, and it's in there, but it's not a good salt. It can interfere with later reactions if you're going to be doing restriction enzyme digestions or you're going to be doing something with your DNA. Cesium is actually able to inhibit later reactions, so we need to use another salt, and we're going to use ammonium sulfate. We're going to use ammonium acetate because sodium is a much more friendly salt to replace the cesium in binding to the negative charges on the phosphate backbone of the DNA. So we're going to add sodium acetate, and we're going to bring it up to a final concentration of around 66% ethanol. DNA has a minimum solubility around 66 to 70% ethanol. So by adding some sodium acetate and basically 2 volumes of 100% ethanol to every 1 volume of DNA solution, we're able then to precipitate out the DNA. So it becomes insoluble, and by centrifuging, you will make it pellet, and then you can get rid of the salt. So in this case, we've added sodium acetate, so the sodium is now with the precipitated DNA, and our cesium will be in solution. so we can discard the supernatant, get rid of the supernatant, and then resuspend it in water because you've purified your DNA. We're going to be actually doing this procedure twice to make sure that we've gotten rid of all the cesium away from our DNA. And following precipitation and bringing and resuspending in water, you want to know what are the results of your reaction. So you're going to take the absorbance of your DNA, and that's going to allow you to measure the concentration of your DNA and what the purity is of your DNA.